As we have discussed previously, eukaryotic chromosomes reside in the nucleus. So, majority of DNA which we can find in eukaryotic cells is inside of nucleus. However, some DNA can be found in the organelles, particularly mitochondria and chloroplast. The DNA and inheritance of the genes located on this DNA follows different patterns than inheritance of genes which are located in the nucleus, and we refer to this inheritance as a cytoplasmic inheritance. So cytoplasmic inheritance refers to mitochondrial and chloroplast inheritance. Now, we will not actually talk much about the structure and function of mitochondria and chloroplast. You discuss this in great detail in General Biology 1. Only thing I would like to point out is that, as you know, mitochondria can be found in most eukaryotic organisms, while chloroplast you can find predominantly in plants. Mitochondrial DNA, I am already focusing more on mitochondria, and that's what we will do in this chapter. We will predominantly discuss mitochondrial DNA and inheritance of mitochondrial genes. So, mitochondrial DNA encodes some polypeptides which are used within the organelle, as well as some RNA, ribosomal and tRNA. In the mitochondria, there is over 900 structural proteins and enzymes. You know that mitochondria is a factory which produces energy in cells, so there are many enzymes present, and most of these enzymes are actually encoded by nuclear DNA. So mitochondria encodes only few of them. Mitochondria and chloroplast resemble a lot the prokaryotic organism, particularly bacteria. And actually, the endosymbiotic theory proposes that both of these organelles were once free-living bacteria that become internalized by eukaryotic cells, and over the time they lost several genes, they become symbiont. They started to live in symbiosis with the organelle, and it was so close that they didn't need certain genes, so they lost them, or some of the genes were actually transferred to the nucleus. So what supports this theory? If you look at mitochondria and chloroplast and compare them to bacteria, you will see that they are very similar in size and structure. Keep in mind that mitochondria and chloroplast possess own ribosomes, so they encode ribosomal RNA, and there are ribosomes found within this organelle. In addition, if you treat cells with antibiotics that inhibit protein synthesis in bacteria, however, do not affect the protein synthesis of eukaryotic cells, these antibiotics will inhibit protein synthesis in mitochondria and chloroplast. So that's a quite strong evidence. However, the strongest evidence came from DNA sequences. Once DNA of the mitochondria and chloroplast was sequenced, it clearly demonstrated that mitochondria and chloroplast DNAs are more closely related to genetic sequence of bacteria than they are to the cells, to eukaryotic cells where they are found. Mitochondria and chloroplasts are found in cytoplasm, and during cellular replication, the organelles are distributed into daughter cells during cytokinesis. As you recall, cytokinesis does not follow such a precise mechanistical steps. It's not such a precise mechanism as is division of the chromosome. So we will talk a little bit how the mitochondrial DNA is inherited. First thing, however, which you have to realize is that there is uniparental inheritance of mitochondrial DNA in animals. In animals, including, of course, mammals, we are animals, mitochondrial DNA is inherited exclusively, almost exclusively from female parent. For a long time, it has been suggested that this is because of different distribution of cytoplasm in eggs and sperms. So we know that sperms have only small cytoplasm and only few organelles. However, recently it has been shown that few mitochondria, paternal mitochondria, can be transmitted from sperm to fertilized eggs. And these paternal mitochondria, which are transmitted to eggs, are actually selectively eliminated. So they are selected against. The cells initiate autophagy, which eliminates paternal mitochondria. The precise mechanism and reason why this happens is not completely understood, particularly because in some plants, actually, and in gymnosperm and angiosperms, actually mitochondria and chloroplasts are inherited from both parents. Now, before we talk about the replicative segregation, the mechanism by which mitochondria are distributed into daughter cells, I want you to realize that individual cells may contain many mitochondria. You can see it in this picture where the nucleus is stained in red, right here, and the mitochondria are stained in green, and in yellow is staining of the mitochondrial DNA. So what does it tell you? What does it show? That mitochondria, each cells contain many mitochondria, and each mitochondria can have several copies of mitochondrial genomes. If you add it up, it actually comes up to the fact that individual cell may contain from around hundreds of organelles with numerous copies, which come up to hundreds to thousands of copies of mitochondrial DNA. That's why when you try to recover DNA from fossils, for example, or from long time frozen organisms, you usually analyze mitochondrial DNA because there is more mitochondrial DNA copies than there is nuclear genome copies of genes. We are referring to the gene copies. So now let's talk about what is replicative segregation, heteroplasmy, and homoplasmy. So we already said that one cell can have many mitochondria, and each may carry several copies of mitochondrial genes, which will result in hundreds to thousands copies of mitochondrial genes within one cell. Now, it's very easy to imagine that that random mutation may take place in some of these organelles, in some of mitochondria, and afterwards the cell will be like this cell, two cells outlined here. It is offsprings of one divided cells. So what can you see in here? That there are different mitochondria. Let's pretend that mitochondria which are outlined as a blue are those which carry mutation, random mutation, and those which are outlined in red carry wild-type mitochondrial DNA. So these cells which carry mutated and wild-type mitochondria in regards to the DNA which they have are called heteroplasmy. So heteroplasmy is actually referring to the occurrence of two distinct varieties of DNA within the cytoplasm of the single cells. Now, when such cell, which this heteroplasmic cell divides, you know that the division of cytoplasm is random. And this random distribution of the different organelles into the two progeny cells is called replicative segregation. So replicative segregation is outlined right here. So during replicative segregation, which refers to segregation of the organelles due to cellular replication, since it is random, this replicative segregation can yield different offsprings. Now, let's look in this line here what kind of offsprings we obtain and how the mitochondria were distributed into daughter cells. So heteroplasmic cell can actually give rise to daughter heteroplasmic cells. This is heteroplasmic cell, these three are heteroplasmic. So they can give rise heteroplasmic cells. However, sometimes what might happen is that by chance the mutated mitochondria are segregated right here into one cell, or the wild-type mitochondria are segregated into one cell. Those cells in which all organelles are genetically identical are called homoplasmy. So this represents homoplasmy. So this is homoplasmy and this cell is homoplastic. Okay. So now, when replicative segregation takes place in somatic cells, it might actually create a variation within a single organism. So different cells may possess different proportion of mutated or wild type sequences, meaning a cell containing organelles with different mitochondrial DNA. When replicative segregation takes place in the germ cells of heteroplasmic donor, this may generate different phenotypes among the offsprings. Mutation in mitochondrial DNA can result in certain disorders and diseases. The first mutation of mitochondrial DNA has been studied in yeast and in Neurospora, two genetic model organisms. I will not discuss those in details. You can actually read it in the book. What I would like to point out is that there are several human diseases which are caused by the mutation of mitochondrial DNA. Many of these diseases are due to the mutation of gene encoding enzymes involved in electron transport during the generation of ATP. One such mutation is outlined here. It's myoclonic epilepsy and red fiber syndrome, which actually is only manifested if more than 90-95% of the mitochondrial DNA carries the mutation. Another mutation which we can define in humans, which is caused by the mutation of mitochondrial DNA, is Leber hereditary optic neuropathy, and it's also results from the mutation of the gene that encodes electron transporting proteins, and it's linked to the loss of vision in the middle age, and it can have kind of severe outcome for the complete loss of vision. Now there are another diseases such as neurogenic muscle weakness, retinitis pigmentosa, ataxia, which are caused by the mutation of mitochondrial DNA. Now in plants, mutation in mitochondrial DNA can actually cause cytoplasmic male sterility. Let's now examine very briefly mitochondrial genome. In most animals, entire mitochondrial genome exists as a single highly coiled circular DNA molecule. Also, there might be several copies of this circular DNA in the mitochondria. So each mitochondria can contain multiple copies of the mitochondrial genomes. Now, one thing which I want you to realize, mitochondrial DNA originated from bacteria, and they resemble more bacteria than eukaryotic nuclear DNA. And therefore, and indeed, mitochondrial DNA do not associate with histones. Mitochondrial DNA do not have histones. They actually interact with proteins. However, these proteins are only histone-like proteins. They are not histones, okay? Now, mitochondrial genomes are small when you compare it to the nuclear genomes, and also you cannot judge based on the size of genome on the number of genes which it encodes. Still, this difference is so great that we can easily say that the number of genes encoded by mitochondrial DNA is smaller than the number of those which are encoded by nuclear DNA. The most genes encoded by the mitochondrial genome are those linked to respiration and oxidative phosphorylation, transcription, translation, and RNA processing. Now, in this picture here, you have outlined human mitochondrial DNA. You see it's a circular, double-stranded. It has two strands, outer strand, which is heavy strand, and inner strand, which is light strand. It's asymmetric. The composition of these two strands is different. Heavy strand contains more guanine nucleotides than the light strand, and it encodes both ribosomal RNA and most of the tRNA and the genes. What I want you to look at is this D loop. D loop is the size where DNA replication initiates. It's the origin of replication. Now, one thing and also the last thing I want you to recall is, or remember, is that organization of mitochondrial DNA is highly economical. There are very few repetitive DNA sequences, and there are very few non-coding nucleotides, those which we found quite often in nuclear DNA. So at the end, some interesting facts. We are all interested in aging. Well, maybe not you, you are still young, but everybody who crosses over the bridge over the 40 and 50s, the aging is a big question. So the symptoms of many human genetic diseases actually linked to mitochondrial DNA mutations appear in middle age or later. And it has been postulated or hypothesized that this is relevant or related to decline in oxidative phosphorylation. So when we age, our oxidative phosphorylation slows down. So why does it decline with age? The possible explanation is that during age with aging we accumulate damage in mitochondrial DNA, and this damage can be due to deletion, base substitution, mutation, or so on. There is one example, for example, 55,000 base pair deletion in mitochondrial DNA can be found in individuals after age 40. It starts to appear. You usually don't see this deletion in younger people who are younger than 40 years. And this deletion actually can start to appear also in brain tissue after 75 and older, in people who are 75 or older. So what is it? What is that relationship between mitochondrial DNA and oxidative phosphorylation? You know that during oxidative phosphorylation, many free radicals are formed. And since mitochondrial DNA is located right in the center of the action, it's physically close to enzyme that actually take part in oxidative phosphorylation, generate free radicals. It has been postulated that mitochondrial DNA can be more prone to oxidative damage than nuclear DNA.